biology. I already told you the questions which come in the exam, and the Viva usually is based on these questions. So you have another DNA extraction experiment, so I expect some questions to come from there in the Viva. So you do DNA extraction from cauliflower mainly. So the first question which may be asked you is that why do we use cauliflower for DNA extraction? And what is your answer for this? The answer is that cauliflower has a good quality DNA. What is a good quality DNA? Is the DNA which has much less of unwanted secondary metabolites and phenolic components. That means what? That those have much less contaminants present in them. So cauliflower we use. Why? Because it has much larger number of cells per unit and they are dividing cells, so more DNA, and secondly the DNA present is of good quality. The next question which is related is that why you usually freeze this before and in between you are keeping your homogenate cold? And what is your answer for that? Is that this reduces the enzyme activity of enzymes, especially the nucleases, which will break down the DNA, and you will not get any recovery if you don't use the cold environment or maybe the frozen cauliflower. Then the another one which comes is what is a good quality DNA? Now you know this already. The answer to this is that which has much less contaminants present. And what are the contaminants? Are polysaccharides and phenolics, right? Because what they will do is they will co-participate with DNA, and when we precipitate DNA they will also precipitate, and that will not give you the good result. Similarly, the high quality DNA has high molecular weight and it has a higher A260 to 280 ratio. So if anyone asks you about high quality DNA, so you need to tell them these two things. One is high 260 to 280 ratio, and the other one is no contaminants or much less contaminants. Then the next question which you may be asked is that what are the 3 steps in extraction of DNA? Then what 3 steps you will tell? First is cell membrane and cell wall lysis. You break down by grinding. And second one is that the DNA has come out. Now this stabilizing the DNA in the extractant, and third is precipitating and washing the DNA. So maybe then we can go in details, but the first preliminary question becomes what? What are the 3 steps for DNA extraction? So the cell membrane and lysis you can explain, but then how do you stabilize this DNA? The question which is very popular and which will be asked, the DNA, of course, it has come out of the cell. What are the methods to stabilize the DNA? And before answering this, you must know why it is getting destabilized. I told you one already. It has nucleases which will degrade the DNA, and also the DNA is present in the cell as nucleoproteins in the chromosomes. So we need to remove them from nucleoproteins. So we have to do the disassociation with the nucleoprotein, and then we need to remove the contaminants. So the things which you need to do in stabilizing DNA is first of all DNA's activities, you have to remove that. Then you have to disassociate this from nucleoproteins, and then you have to remove the contaminants which are present. So then, sometimes to confuse, I was just writing these random Viva questions, and suddenly, after having talked to you so much of complications, sometimes I don't get answers. So I suddenly come back to DNA structure, and I might like to ask, who discovered DNA? You know who discovered DNA? Is Misher in 1869. And then back to the DNA extraction again, because the Viva questions are based on the practical you are doing. Then how do you protect your DNA from action of nucleases? How would you protect your DNA from action of nucleases? Is because you are adding the chelating agents, EDTA, And what is this chelating agent doing is, it is removing the metal ions which are required for the nuclease activity. So first here is you use EDTA, that is the chelating agent that will require the metal ions, and so the nuclease will not be able, and the NaCl presence also helps you in removing the activity of DNases. So using the cold environment, using the chelating agents and NaCl, that helps you in stabilizing the DNA and removing the nucleases activity, meaning preventing the cell from degradation of DNA. The other one is since the DNA is present in chromosomes, how do you separate it from nucleoproteins? Then how do you separate? Because when you are performing the experiment, you are looking at the protocol, and in the protocol you have several steps, and in these steps where you are disassociating is that you are using SDS or phenol or maybe enzymes, and these three compounds in your steps, you will see they are for disassociation from the nucleoprotein, and the job is done better if you have alkaline environment and high salt concentration. So if you check your steps, you can confirm that you are using SDS, phenol and other enzymes to disassociate the DNA from nucleoproteins, and you do this in alkaline and high salt concentration. Sometimes that gives you the confirm that the spool you have got is not contaminated by polysaccharides and phenols. You will do gel electrophoresis, and if in the gel electrophoresis it shows one clear band of DNA, that means there is no contamination, because polysaccharides and phenols, they have different properties and they will separate differently. So now if I ask you to tell me the concentration of DNA in your sample or in your precipitate which you have isolated, how will you do that? How would you know the concentration of DNA in your sample you have extracted? Is you have done in the class, that is diphenylamine methods, DPA method. Then I might ask you, what is the principle for DPA method for DNA estimation, diphenylamine, and you know the answer for this. What is the answer? The answer for diphenylamine assay for DNA concentration estimation is that when DNA is treated with diphenylamine under acidic conditions, it forms a blue compound, and that can be measured by the spectrophotometer at 595 nm. Right? So DNA in under acidic conditions, it will react with diphenylamine to form a blue compound that you can measure by spectrophotometer at 595 nm, and that is how you will know the concentration. So another question specifically that the DNA is interacting with diphenylamine, or which part of DNA, meaning nucleotide, base, sugar or phosphorus, which is reacting with diphenylamine. And here you have to say this is the deoxyribose sugar which is acting, and that is why sometimes it is said that the DPA test is not very specific. So what is the principle of estimation of DNA by DPA is, again, as I told you, the DNA is treated with diphenylamine under acidic conditions, and it forms a blue compound which can be measured by spectrophotometer. So the next question which comes here is that why ethidium bromide method is not used in nowadays, though this method was good, but why it is not used? Because if you remember, in mutations we studied, and it is carcinogenic, it causes the cancer. Next question, that is question number 28. What are the limitations of DPA method? The main limitation here is that it lacks sensitivity if your DNA concentration is greater than 1 microgram. So it lacks the sensitivity there. And how you can improve the sensitivity of DPA method is by is by increasing the incubation time. First of all, you usually incubate this for 10 minutes. If you incubate it a bit higher, you will be able to make it more sensitive. And then secondly, by using a better spectrophotometer. So this way you can improve the sensitivity of DPA method. So these were 29 questions till now, but still it's already time and I have to break this recording again. I will record for another 5-10 minutes from Mendel's laws and share that with you, but I think I have to say bye for now. You prepare these questions and I will very quickly give the questions. So it is done for DNA estimation and DNA extraction. Now I need to have some questions for interactions. Maybe I'll do that very quickly before the Viva, actual Viva happening. Okay, bye for now. Thank you.